Hi, thank you for joining us for the 7th FKMCD Oxitec Public Educational Webinar. Our topic tonight is Human Health and Oxitec, the Safety of Oxitec Technology. Our panelists this evening are Andrea Leal. Andrea is having, there's a problem with her webcam, but she is with us and you'll be able to hear her speak even if you cannot see her. Andrea is the Executive Director of the FKMCD. I'm Meredith Finsom, Oxitec's Head of Public Affairs and a Florida native. Kevin Gorman is our Head of Field Operations, and Nathan Rose is Oxitec's Head of Regulatory Affairs. We're hosting this series of public educational webinars to share information with residents of the Florida Keys and provide forums to answer questions. All webinars are open to everyone. All webinars are recorded and made available for everyone after the event. These are public on our websites. All questions will be answered. We may group similar questions together. If time runs out, we will accept questions in writing via florida@oxitec.com. Questions and answers will be published in writing after the event with links to external resources. We will not identify who is asking the questions. We want you to feel comfortable to ask anything at all that you are curious about, but we do ask that questions pertain to tonight's topic. We'd also like to flag for you our upcoming webinars. We're going to have at least one a month through March, probably beyond. But next month, we are featuring a virtual tour inside Oxitec Labs. Those are in production now in the UK, in Marathon, and in Brazil. And it should be a really exciting webinar next month, and we really hope you can join us. That'll be November 18th. As far as our agenda this evening, first we'll look at why now, health, economy, and the environment. We will look at disease transmission by the invasive Aedes aegypti mosquito, the current options for mosquito control, the benefits of Oxitec's targeted biological control solution, and the safety of our male mosquitoes. We'll look at regulatory findings, and then we will spend the second half hour of this webinar answering your questions. All of the documentations, resources, references, and other information about our project and all the materials from these webinars, including the recordings and fact sheets, they are on the recently jointly launched FKMCD Oxitec project website. It's called keysmosquitoproject.com, and we invite you to visit that. Now I'll turn it over to Andrea to begin our presentation.
Thanks, Meredith. And again, I apologize that you guys don't get to see my smiling face this afternoon, but unfortunately I'm having some technical difficulties. So I guess I'll start out talking a little bit about why the district specifically is looking at this technology and similar technologies. Back in 2009 and 2010, we had an outbreak of dengue fever in Key West, and we had just under 100 cases of dengue. At that time, there really weren't a lot of tools that were specific for the mosquito that transmits that disease. And we've really been doing our best here at the district to control Aedes aegypti, and that's the mosquito that causes dengue specifically. And we didn't see cases for about 10 years. But after that first outbreak in 2009 and 2010, we really started looking around at what new technologies are out there and what can we do to, really enhance our Aedes aegypti control program. Fast forward to this past year, and we've seen 67 confirmed cases again of dengue fever in the Key Largo area. So we know that we have these Aedes aegypti mosquitoes around, and the district is doing really all that we can in our power to control these mosquitoes. But we are seeing more and more cases of introduction of these kind of diseases into Florida. And unfortunately, when we have these mosquitoes in certain levels, then you'll start seeing local transmission occurring. So, this is one of the biggest reasons that we're focusing on these new technologies that are coming out. How can we do our best here at the district to protect people out there? Not only are we worried about dengue, but we saw introductions of Zika in Miami-Dade in 2016, as well as chikungunya. We've heard of yellow fever outbreaks occurring in South America on a regular basis. And on top of all of that, we are starting to see insecticide resistance in our local mosquitoes. So we have a very limited number of mosquito control tools that we can use that still work for these specific mosquitoes. And unfortunately, that toolbox keeps shrinking. And so, again, this is why we're looking at this Oxitec project, wanting to increase our toolbox, really trying to find the best ways to control these mosquitoes and protect, the folks here in Monroe County. Additionally, as we're looking at new technologies, it's really nice to see that we can focus on specific Aedes aegypti species control. So, one of the things that the district does, a lot of you are familiar when our fog trucks go by, that's putting out a general insecticide, and could have some non-target effects. Now, again, we apply at times where we're trying to minimize those effects, but that's one of the nice things about the Oxitec mosquito specifically is that it is there for Aedes aegypti control only. We don't have to worry about any other environmental impacts, any other species being impacted. And, looking at this tool, we've seen, from the EPA as well as local state wildlife offices that, they're very interested in us moving forward with this trial. Next slide, please. So I mentioned a little bit about the diseases that this particular mosquito can transmit. We're here in the Florida Keys, we've seen locally acquired cases of dengue, but just to our north, they've seen basically almost all of these, and they're all transmitted by this one mosquito. This one mosquito, unfortunately, is also very difficult to control. She likes to live in and around homes, under homes. All it takes is a tablespoon of water, basically, to create more of these mosquitoes. So she's very difficult to control. And really, our focus is to make sure that we're not seeing these diseases. I know that, again, we've had 67 cases up in the Upper Keys this year. Majority of people were experiencing these symptoms of dengue, headaches, fever, rash, joint pain. It can be very, very painful, and it can last for weeks at a time. Next slide, please. And again, we're focusing on one mosquito, the Aedes aegypti mosquito. And this particular mosquito is an invasive species. It was not found here, was brought over in the 1600s. With it came, a number of these diseases. So, we're not looking at really controlling those mosquitoes that are native or part of the local ecosystem. Down in the Florida Keys, salt marsh mosquitoes specifically are a big part of our ecosystem, especially the mosquito larvae themselves. So out in the swamps, you've got other insects, fish, birds that'll feed on those salt marsh mosquitoes, but you don't have that same niche as for the Aedes aegypti. They're found in discarded tires where you're not going to find predators. They're found in your gutters. They're found in 5-gallon buckets. So really focusing only on that invasive mosquito species. And that's one of the great things about this technology. Next slide, please. So we really do have a very robust mosquito control here in the Florida Keys. If you compare us to, throughout the nation, we do have one of the highest budgets, and we do a lot of different control methods currently for Aedes aegypti. Currently, we're using a number of different insecticides, but again, when it comes to those broad-scale adult mosquito control products, we are starting to see resistance, and that is becoming an increasing problem for us. What you'll see a lot of our response for Aedes aegypti control is aerial bacteria spraying. So we did this about once a week in the Upper Keys throughout the entire summertime here. And it's basically putting out a bacteria that the mosquito larvae then ingest, and it kills mosquito larvae. It's another great tool for us to use because it is specific for mosquitoes. And so we don't have any other non-target effects. It's great for the environment. The problem with that is, looking at our response recently to dengue, the district has spent, over $1 million of our budget responding, for the last 6 weeks, just in that area. So again, we really need to find more tools to help us control these Aedes aegypti mosquitoes. And that's really the biggest reason that we're looking at this technology and excited for this upcoming project. And I think with that, I'll go ahead and turn it over to Kevin.
Great. Many thanks, Andrea, and thanks, Meredith, for the introduction. For these next few slides that I'd like to run through, I'm going to just talk a little bit about the technology, just recap over the salient points, and talk a little bit about how this technology works. And then I'll hand over to Nathan, who will finish the presentation. This technology is a mating-based technology. So we release male Aedes aegypti, and those male Aedes aegypti then go out and mate with wild female Aedes aegypti. Those females are unable to have female progeny survive through to adulthood. And so all their female progeny die, and with successive releases over successive generations, that population is brought down quite rapidly through no more females being produced. Now, how do we do that? Well, that's where the genetics come in. We are able to produce male-only cohorts from eggs. So these eggs aren't able to produce females, as I've just mentioned. And so we can put those eggs into very simple, low-tech boxes, essentially, which are very easy to deploy, very scalable, and they can go out into the environment with some eggs and some diet, and those males only will come out of those boxes. They'll rear themselves and release themselves into the environment, and then they'll go out in search of those wild females. So it's a biological approach, it's mating based, but it's a simple approach in terms of its operational deployment. And that's where it becomes quite unique, because it's scalable. It's something that could be accessed and used by everybody from a competent vector control authority through to potentially an alternative end user, such as even a residential customer. Once those males go out into the environment and start to mate with those wild females, all the progeny and those males themselves are identifiable with a fluorescent marker. So under a certain wavelength of light, they fluoresce, and that means that all life stages are identifiable, and we can tell exactly how well the strategy is doing. We can tell if we're getting a certain percentage of those offspring parented by our male mosquitoes, and that allows us to maybe change the dose or change the strategy to ensure that we are getting the right level of control. That means that we can be really efficient too. So we're not having any redundant releases. We're making sure that our mosquito releases are nice and efficient, which helps bring down the costs and helps maintain a very healthy strategy. So that's in a nutshell how it works. I would also add that there is no toxin involved. This isn't a chemical insecticide. This is just a benign protein that's overproduced in these female insects that die. It enables us to get really strong effectiveness in a sustainable manner that's not actually prone to resistance development like something with a particular active site might be. And as I mentioned, it's fully traceable in the field. And one other unique feature is that even though we get that multi-generational effect from those male offspring that carry through those subsequent generations, it's also self-limiting because they die out in a half-life type of manner. So once we stop releases, quite quickly, over a period of typically 3 to 10 generations, those male mosquitoes die out, and we end up with a pristine environment, just like it was before we released our mosquitoes, this biological approach, with no ecological footprint, apart from the fact that we've taken away some of those pest Aedes aegypti that could have been transmitting diseases. So biological, sustainable, and no persistence. So why males? Just a quick recap. It's females that bite. It's females that transmit diseases. Males don't have the right mouth parts to bite humans and transmit disease. They feed off nectar and typically flowers, trees, etc., and other sugars. Females need that blood that they get from human biting to mature their eggs. They need the protein from that to help mature their eggs, and that's why it's the females that cause the problems for humans by transmitting a range of different viruses, and Nathan will come onto those in a little while. So how does that self-limiting gene work? There are a number of features here about that self-limiting gene that I've already mentioned. The females can't survive, and those male mosquitoes are unaffected. But the key thing is that this is a benign protein that's produced. The self-limiting gene just produces a protein that enables the cell, that makes the cells go into overproduction, and those cells aren't then producing the proteins that they're supposed to produce to make sure that insect is fit and healthy, and that insect dies, a female dies at a very young aquatic stage, one of those early larval stages, way before it can become an adult and start to bite people, take blood feeds, and transmit diseases. So it's male-only production. It's foolproof male-only production in that sense, in that you don't need any high-tech or any additional ingredients. You only need to rear up the eggs, and male-only cohorts come off. And that enables suppression, as I mentioned, of the successive generations. So 0 females are produced and released as live female adults are produced and released through this technology, and males carry on that genotype and pass it on to their female offspring, helping to increase the performance of the product and ensuring that we get additional kill from each generation of our male mosquitoes. We've released quite a lot of these mosquitoes now. So over the last decade, we've released over 1 billion OX513A, or our first generation mosquitoes, globally in a range of countries and across several continents. And we've also now released probably over 20 million of our male 5034 mosquitoes, which are the ones that I'm talking about today, and that Nathan and will be talking about shortly. That's a lot of mosquitoes over a long time. We've never had any negative impact observed either for humans or the environment. We've done a lot of good over those years, and we've reduced populations in many different regions by very high percentages. Okay, so there's a fluorescent marker. I mentioned it a short while ago. This is a DS-Red2 protein. It is a protein that's used in lots of technologies. And it just simply fluoresces under a specific wavelength of light, so we can bring those mosquitoes in from traps, be those adult traps or be those what we call ovi traps, which collect eggs, and we can hatch them out, and then we can put those insects under a microscope, and we can tell by looking at them and seeing whether they fluoresce or not, whether they were parented by an Oxitec male. And in a couple of those photos that you see there on the right-hand side, there are larvae and pupae, the two on the left-hand side, and there are actually wild-type ones which aren't fluorescing adjacent to the fluorescent ones. So it becomes a very simple way of tracking and tracing these mosquitoes, and enables, as I mentioned, to look at our dose rates and make sure that our performance is efficient, not too high, but also that it's high enough. And it also allows us to look where these mosquitoes go and to follow their pathways and their behavior, and allows us to get really interesting insight into exactly how these mosquitoes are working for us. As I mentioned, it's produced in all life stages, so that's larvae, pupae, and adults, except for the eggs. And males pass on that fluorescent gene to all their offspring, just as they do the self-limiting gene. But whereas only females express the self-limiting gene, both males and females express this fluorescent protein. So genetic integration, because of those males that are surviving, those males are able to mate with wild females, and their offspring then carry some of the background genes, and they survive. Now, they carry the self-limiting gene too, but they don't express that. All the females die. Those males, when they mate with females, they can pass on to half of their offspring those genes. So that's the half-life fashion that it dies out of over successive generations. But some of those offspring, the other half, they can inherit some of the background genes. And that is particularly useful because we can, using that format, we can actually introgress really beneficial genes, such as those for insecticide susceptibility, into that target population. So not only are we then suppressing the population as a whole, but those insects that are left become diluted by their insecticide resistance becomes diluted by insecticide susceptibility, and that allows potentially a restoration of insecticide efficacy, and it allows solid integrated vector management programs to be put in place, really prolonging the tools that vector control authorities such as Andrea's really need so desperately. The EPA, CDC, and others have looked at this level of integration that we get, and they've examined this as part of this EUP application that we've put forward. And they've done a very thorough, comprehensive study on the integration and any effects that that might have. And they've concluded that there is absolutely no risk from this, that it is not likely to produce hybrid vigor, that there's no potential for increased vectorial capacity. But indeed, the potential to dilute insecticide resistance is indeed there. And so those benefits that we are seeing really don't come with any compromise at all in terms of safety. And this last slide of mine before I hand over to Nathan is really just to remind everybody how common biotechnology is used almost everywhere in public health and everyday life these days. It's not just those GM crops that people know and love so well. It's also aspects of industrial applications, it's food production, it's healthcare, it's environment, be it vaccines, be it alcohol production or fermentation processes, it's textiles, clothing. There are numerous ways of in which biotechnology is extremely beneficial to modern living. And I'm sure that everybody uses many of these products constantly, whether they know it or not. These are ingrained in our society and have huge benefits for everybody. With that, I will pass over to Nathan, who will take you through the remainder of the slides.
Okay, thanks, Kevin. So I'm going to briefly talk about here the scientific assessment process that was undertaken by the Environmental Protection Agency in the US that granted approval for this project in Florida. So they undertook an exhaustive scientific review process. It took over a year. It included 70 plus substantial studies, 4,500 pages, including 2,500 pages of scientific peer-reviewed literature, which they assessed as part of this risk assessment process. It also involved other agencies, including the CDC, as Kevin just mentioned. And it also took into account extensive public comments. And as part of this assessment process, the EPA also made sure that they responded to all of those public comments. And all of their risk assessment documents, their memoranda on various aspects, their responses to public comments, and so on, are available through the regulations.gov website, and there are links to all of those documents that can be found through the Oxitec website as well, which Meredith mentioned at the start. So if anyone is interested in seeing how they carried out this risk assessment and seeing their thought processes as they looked at this mosquito, it's worth taking a look at some of those documents. The data requirements were extensive. We talked in the previous webinar in September about some of the environmental risk assessments that were carried out. So I won't go into detail of those again, but I encourage you to have a look at the September webinar recording or the fact sheet that came out of that. When it came to assessing the impact on human health, they looked at several parameters. Trait penetrance is important, and that is really making sure that all of the female mosquitoes die as expected. And they confirmed, looking at all of the data provided, that 100% of female mosquitoes die when they inherit the self-limiting gene. They also looked at a range of toxicity endpoints, so oral toxicity, inhalation toxicity, toxicity to the eyes or to the skin. They looked at allergenicity as well, and in all of these concluded that there was no risk of toxicity or allergenicity as a result of the releases of these male mosquitoes. As Kevin just mentioned as well, they looked at potential impacts on the vector competence of the wild mosquito population, so the ability of the wild mosquitoes to transmit disease, and again concluded that there was no risk of increasing vector competence through the release of these mosquitoes. In fact, the release of these mosquitoes will lead to suppression of the wild population, and thus a reduction in disease transmission is expected. And then they also looked at a wide range of characterization studies of the mosquito itself, so ensuring that it was fully susceptible to relevant insecticides, which it is, looking at its response to tetracyclines, looking at the stability of the genetic traits which have been introduced, looking at the field data from successful field trials in other places, particularly in Brazil, and then a range of other aspects as well. And the EPA also reviewed the complete standard operating procedures supplied by Oxitec for the production of the mosquito and also for the release and monitoring of the mosquito in the field as well. So a very extensive scientific data analysis carried out by the EPA, and at the end of this, they concluded that there was no risk either to human health or to the environment as a result of the releases of these mosquitoes. We also had the CDC look at some aspects of the risk assessment, which we've just mentioned, but the CDC will actually continue to be involved in this project. They have intended they've written to communicate their intent to collaborate with FKMCD on various aspects of this project, including the technical evaluation to make sure that the technical outcomes of this project are properly evaluated and the outcomes are statistically sound and can actually give a clear picture of how well this technology works in the field. So we look forward to having the CDC involved as we move forward with this technology. I'm also going to spend a few minutes talking about another aspect of the mosquitoes. So Kevin's talked already about the genes that are introduced, how they produce proteins which are safe and non-toxic. But I'm also going to talk briefly about how the mosquitoes are actually made, first in the UK, and then what happens once they're actually shipped over to Florida. And I'm going to spend a little bit of time here because I think this will be helpful just to understand, firstly, how they're made, but also how they are actually safe throughout this production process. So what happens in the UK is that we take some of the eggs from this OX5034 strain, we hatch those into larvae, and a small amount of tetracycline is added to the water that those larvae grow in, so that in the UK we can produce both males and females of this particular Aedes aegypti strain. And that's important because we want the males and females to be able to mate and to produce more eggs, so we can then ship those eggs to Florida for release in the field. So that tetracycline is important because it helps to turn off the self-limiting gene in females at those very early larval stages, and that allows them to survive through to adulthood. And so the tetracycline is used only at the larval stages of mosquito production and only in the UK. After they turn from larvae into pupae, there is no more tetracycline. They are washed extensively and moved to clean water. And then those pupae are then moved into cages where they will eclose from pupae to produce the adults. And then those adults can mate with each other. The females will take some blood meal, and then they can lay eggs. Those eggs are then surface sterilized with sanitizer, which is actually significantly stronger than hospital-grade disinfectant. And then after that surface sterilization, they are packaged. And then those packaged eggs are what will actually be shipped to Florida for release in the field. At every stage of this process, there is extensive quality control to make sure that they are produced appropriately, to make sure that the self-limiting gene is functioning, and also to make sure that the males are performing as expected. So the tetracycline is used only in the United Kingdom, and that's only in a very small quantity to produce those eggs. To produce all of the mosquito eggs for this entire project in Florida, Oxitec will use less than 5 grams of tetracycline. So after those eggs are packaged, they are then shipped from the UK to the US. Once they get to Florida, they are then taken into the field where they're put into these mosquito rearing boxes. And we add water, we add some diet, and we put that box in the field. So that's placed in the field in an out-of-the-way location by an FKMCD or an Oxitec operator. And after a few days, those eggs will have hatched. They will develop into larvae, and all of the female larvae will die because there is no tetracycline in that box. They cannot survive without any tetracycline present there, whereas the males can. And so the males will then develop into adults. They will escape from the box, fly out into the field, and go and find wild females to mate with, as Kevin described earlier. So there's no tetracycline in that box. There's no tetracycline used at all, actually, in the US to produce the male mosquitoes for release. So I'm bringing this up just because I'm aware that there's been a suggestion that the use of tetracycline might bring potential risks of spreading antibiotic-resistant bacteria in the US environment. And this is something that has been looked at extensively by the EPA, previously by the FDA when they looked at the earlier version of the Oxitec mosquito, and also by 8 state agencies within Florida that actually assessed this project. And all of these have concluded that there is negligible risk that these mosquitoes would contribute to the spread of antibiotic-resistant bacteria in the US environment. So the EPA concluded that, the FDA did previously, and the Florida state agencies did the same. And a lot of data went into this conclusion, and all of the details of how they came to that conclusion can actually be read in the documents that I referenced earlier. So it's worth taking a look at that because they looked at the production process in detail as part of this risk assessment. I'm aware that there has been a suggestion from a local citizen that Oxitec should carry out even more testing along these lines. And I'd actually like to address that clearly here. With EPA, the FDA, and 8 Florida state agencies having looked at this particular question, and that's 10 agencies in total, but that represents dozens of independent scientists. They've all examined this carefully and concluded that there is negligible risk that these mosquitoes could contribute to the spread of antibiotic-resistant bacteria in the environment. And so there, after all those conclusions, there is actually no basis for further testing of this particular question. I'd also just like to put this in context. So I mentioned in the UK about 5 grams of tetracycline will be used in total to produce all of the mosquitoes for this particular project. That's equivalent to about 2 therapeutic courses of tetracycline for humans to treat an infection. In the US, by contrast, every year, doctors and emergency departments will prescribe about 47 million courses of tetracycline antibiotics for infections that don't need them. There are about 4,000 tons of these supplied to livestock and pets. And in Florida alone, the EPA has authorized close on 400,000 pounds of antibiotic spraying, oxytetracycline and streptomycin, for citrus farms to control a particular disease of citrus trees. And so just looking at this in contrast as well to what will actually be used in another country to produce these mosquitoes, that goes into this risk assessment as well, leading to this conclusion that there is negligible risk of these mosquitoes spreading antibiotic-resistant bacteria as a result of their release. I'd also like to talk, moving on from that, about the mosquitoes themselves and what they've done previously in the field. So we've talked about this in much more detail in one of our previous webinars, and the link to that is on our website. But we have actually released these mosquitoes in several countries. The first version of the mosquito, OX513A, for about 8 years. And then in 2018, we had the first pilots of this OX5034 mosquito in Brazil. And all of these have had successful suppression of the wild mosquito populations. And with the OX5034 mosquito in Brazil in the last 2 years, we've had very successful results. 96% suppression of the wild mosquito population in those trials, in the first trial in a city called Indaiatuba in Brazil. And last year, we also trialed this just add water release of the mosquito eggs and achieved a very similar result also in Brazil. And we will be continuing those releases in Brazil starting in just a few weeks' time, again in São Paulo state in Brazil. So in a country where diseases are endemic, where dengue is a major problem, this has been a very successful technology in actually being able to control this vector of dengue, of Zika, of chikungunya, and so on. So it's an important tool that can be used by public health authorities there to actually be able to protect people from these diseases. And that's a really important thing to note here, that this is a mosquito that is really designed to combat the wild mosquito, which is such a dangerous part of life in the tropics, where it can transmit all of these diseases. And then finally, just to summarize what I've mentioned previously, the project that we're carrying out in Florida has been vetted very extensively by the EPA, and in Florida by multiple state departments and bureaus, the Florida Department of Agriculture and Consumer Services, which is responsible for all trials of new mosquito control tools, the Florida Department of Environmental Protection, the Florida Fish and Wildlife Conservation Commission, and the Florida Department of Health have all weighed in approving the permit for this project. And just a further endorsement on an even larger scale, internationally, the World Health Organization in 2016 recommended Oxitec mosquitoes for pilot deployment to fight what was at that time a growing threat posed by the Zika virus, particularly in South America. So this is a technology which has been extensively studied, tested, and approved by multiple agencies at the state level, at the national level, and even at the international level as well. So just to summarize what we've said, there is an increasing disease threat in Florida, in Monroe County, as has sadly been seen this year with the dengue outbreak in Key Largo. It's important to have good tools to protect against this invasive mosquito species. And Oxitec's male mosquitoes are a tool that can really help with that. And they are themselves safe. They pose no risks to human health. Because they're male mosquitoes, they cannot actually bite or spread disease themselves. And this is backed up by a long track record of releases of these mosquitoes worldwide with no negative impacts. So with that, I'm going to hand back to Meredith, and thank you.
Thank you, Nathan. So we'll start our Q&A now. And just to remind everybody, if we do run out of time, you can send questions about tonight's webinar topics or other items related to our project to florida@oxitec.com. And we also invite you to visit our websites. We have the oxitec.com/Florida webpage and the recently launched keysmosquitoproject.com website, and a lot of great information there. we don't have that many questions right now. So those of you watching, if you'd like to add some, we invite you to do that. But we'll start with this first one. Nathan, it's for you. And the question is, can you talk through some of the concerns raised by a certain physician in the Keys relating to tetracycline? Why hasn't the EPA or any of the other regulators taken up his concerns?
Okay, thanks for that question. So this is some of what I've just been talking about. The suggestion that the release of these male mosquitoes might contribute to the spread of antibiotic-resistant bacteria. That is obviously a real concern, to make sure that antibiotics continue to have use and that they can still be protective for humans against the bacteria which cause disease. So that's something which we definitely take seriously, and which is also taken seriously by the regulatory authorities. The concerns alluded to here have been raised with the EPA and also with the state regulators. They were also previously raised with the FDA. And they have actually been responded to at great length by the regulatory authorities. So the EPA, in their response to all of the public comments that they received, devoted quite a lot of that document to addressing this specific issue. And as part of that risk assessment, they looked at the production processes that I ran through a few minutes back. They looked at how antibiotic resistance actually works and how it's spread and how it's maintained. And they also looked at what the male mosquitoes that would be released would also be doing in the field. And all of those different aspects together played into their risk assessment and their conclusion that there was no risk of these male mosquitoes contributing to the spread of antibiotic-resistant bacteria by their release in the field. So a much more detailed account of that and the details of the EPA's argument and all of the scientific literature which they cited making those conclusions is available through the response to public comments document, and there is a link to that on Oxitec's website. But just to say, this is definitely something which has been looked at, which has been taken seriously. And as part of that, they have looked at all of those comments, but they've also concluded that there is no risk. And therefore, they have also said that there's no need to carry out further testing, having looked at all the different aspects which contribute to that risk assessment. So I'm hoping that that answers that question. It is definitely something which has been taken up by the regulators, looked at, and concluded there is no risk there.
Thanks very much, Nathan. The next question is for you also. And the question is, has Oxitec or any government agency or independent body ever documented any negative effect to human health anywhere it has released any insects? And I'll just add before you begin to answer, Nathan, for the people watching, those of you in the Keys hear about our mosquitoes, but we do have agricultural crop pests that we work with as well.
Okay, so the simple answer is no, but I'll flesh that out a bit further. So Oxitec has released its mosquitoes in a number of countries over the last 10 years, initially in the Cayman Islands and in Brazil, in subsequently larger trials in both of those places. And the results of those trials have been published in the peer-reviewed literature. And so links to those papers are available on the Oxitec website. And then after that, trials also in Panama, where we had successful suppression of the wild population there too. And then after that, we actually had approval by the Brazilian authorities to be allowed to release these mosquitoes anywhere in the country in Brazil. Following that, we carried out larger scale releases in a couple of cities where we had substantial control of the wild mosquito population. That was all with the first generation mosquito. In 2018, we pivoted to the second generation mosquito, which has a variety of benefits that we've described in this evening's presentation. And again, we've had 2 years of releases of that in Brazil. No negative effects there either. And that's also recently had biosafety approval for the whole of Brazil to release that anywhere. So in all of these projects, in Brazil, in Panama, in the Cayman Islands, in Malaysia, where we've also released these mosquitoes, we've had successful suppression of the wild population, but we've never had any documented negative effects either on the environment or on human health. And all of those projects had extensive monitoring that took place throughout the project, and then also after we stopped releasing mosquitoes, to see if there was any impact on any of those. And in all of those cases, the mosquito has been safe to use and effective.
Thank you, Nathan. Okay, this next question, Andrea, it's for you. And the question is, can FKMCD share more about its efforts to combat the recent dengue outbreaks? Can we expect more outbreaks in the future?
Sure. So the main thing that we do at the district to combat dengue or Aegypti specifically is basically going door to door. So all it takes is a little tiny bit of standing water to create these Aedes aegypti mosquitoes. And unfortunately, without everybody in the entire Keys in the habit of walking around their properties and dumping out standing water, a couple times a week, then we get these pockets of higher numbers of Aedes aegypti. So when it came to what we did in Key Largo, we had a number of inspectors. We were sending inspectors from Key West and Marathon up to that Key Largo area, and they were going door to door, just talking to residents, letting them know what was going on, and then walking around yards and trying to find and dump any standing water, treat any standing water that couldn't be dumped, and really just going through and doing our best to clean up the neighborhood and educate people on what they can do to assist with this. In addition to that, we were flying helicopter missions with larvicide about once a week, depending upon rainfall. And then on top of that, if our traps, we set out extra BG traps, and they're specifically designed to catch these Aedes aegypti mosquitoes. So normally we have about 10 traps that we set out. We added another 10 on top of that to really monitor the population. And if we were seeing numbers that were higher than what we considered acceptable, we did do aerial adulticide missions or truck adulticide missions. So we really were hammering that area for a good 6 to 8 weeks. The really cool thing that I like to say now, though, is, we first started seeing cases that we knew about in, I think, around June. And we saw cases going all the way through to the end of August. But we stopped seeing cases, and we were still having rainfall. We would still, you would anticipate seeing more cases, but really feel the combination between all of the extra testing that the health department was doing, in addition to everything that we were doing, really made a difference. And we've not seen that really in South Florida before. Normally, these outbreaks continue until, there's a weather reason that they stop. So usually November, you'll stop seeing cases. But we're really excited that, all of our efforts and in conjunction with the health department seem to pay off. Now, the biggest issue that I mentioned earlier is all of those efforts cost the district in excess of $1 million between manpower, chemicals, flight times, etc. It is extremely expensive. So it's not really sustainable to do that type of effort throughout the entire Keys. So what we'll be doing in the future is, we're going to be doing more trapping, again, looking at these new technologies to supplement what we can do. And really we've got to just be better at predicting where these things are going to occur. So if somebody would have asked me where my guess would be, I would have never guessed the neighborhood that we saw. But things have changed. There are a lot of rental properties. People are moving around differently. So, it's something that we're really taking very seriously. We want to make sure that, we're doing the best that we can for everybody and doing the best to prevent something like this from happening. We would rather be very proactive. That's always been our approach here at the district, than reactive. So again, that's why we're looking at, what other ways can we utilize to help us in this fight?
Thank you very much, Andrea. Our next question has a couple of parts. And Kevin, I'll start with you, and then we'll turn to Andrea to also help answer this question. The question is, I've heard repeated statements touting Wolbachia as the safer, more effective type of sterile insect technique. How do these two methods compare in the real world? Is Wolbachia even approved for use by rank and file mosquito control? Thank you.
Thanks, Meredith, and thanks for the person who asked the question too. Wolbachia and Oxitec do differ, and I would maybe throw in SIT there as well as another analogous technique. Those three often get compared, I think, because essentially they're releasing males that aren't able to produce viable offspring or viable female offspring. In our case, it's viable female offspring. So in their concept, they do have similarities, but how they achieve that, they are quite different. In the case of sterile insect technique, it's through radiation, and they use radiation to sterilize those males before they're released. In the case of Wolbachia, it's cytoplasmic incompatibility that prevents those offspring from being born. And in the case of Oxitec, we use genetics to give us that effect of no females. But I guess the key difference really is about the operations, because as we saw with Ox513A, where we killed both males and female offspring using genetics, what we found was that we had a very onerous husbandry, if you like, as we reared those males through to release, as we looked after them, did the QC, and then actually had the logistics to get male adult mosquitoes out into the environment. And those other techniques have that same operational burden, if you like. With our technology now, it's an operational switch, and it allows us to do a very simple low-tech release, a no female release without any extra equipment or ingredients, and as such, it becomes far more efficient and scalable. And I think if you take out the questions about about how that sterility or that lack of successful mating is achieved, it's really an operational question, and that's where these techniques differ markedly. In terms of whether Wolbachia is available, Andrea can answer more, but for Aedes aegypti, it's not yet. It's another one of the technologies that's being assessed by EPA. And so if we're talking about the US, it's not available commercially at the moment, but it may well be in some time in the near future. And let's be clear, there are lots of different techniques for controlling insects, but and some of those are very useful for Aedes aegypti, but there's a lot of Aedes aegypti out there, and we need all the help that we can get to make sure that these diseases are kept under control. Andrea, have you got anything to add?
Yeah, I'll chime in. We actually did a very small trial with the use of Wolbachia, and I think it was in Stock Island in 2017. Unfortunately, the project didn't go the entire duration due to Hurricane Irma, but we saw very positive results. So, the district continues to follow all of the other trials that have been going on with Wolbachia, and really have continued to be in contact with the company and are really just awaiting product registration. As I mentioned earlier, we're open to all of these different technologies as long as there's approvals to move forward in these types of projects. And so that Wolbachia is still on the table for the district, And, should project registration occur, then that is definitely a tool that we're going to be utilizing.
Thanks, Kevin and Andrea. Nathan, the next question is for you. The question is, can you talk through what the US FDA's ruling was and how that relates to the technology you want to deploy in the keys? Was their finding the same as that of EPA?
Okay, thanks for that question. So the US FDA assessed the earlier version of Oxitec's technology, what we call our first generation mosquito, or OX513A. That was the mosquito that we released for about 8 years, from 2010 to 2018 in several parts of the world, including Brazil, Panama, the Caymans, etc. And so in 2016, the FDA gave approval for a small trial of that mosquito in Key Haven. And then following that, the FDA was continuing to assess that mosquito for trials in other parts of Florida, including the rest of Monroe County. But while they were still carrying out that assessment, the jurisdiction over these sorts of mosquito products switched from FDA to EPA. So the FDA approved the earlier version of Oxitec's mosquito, carrying out similar kinds of risk assessment to the EPA, and concluding that with that, there was also no risk to human health or the environment. But by the time we came to actually putting in an application for a project with the second generation mosquito that we've talked about here, the one that allows the males to survive while making sure that there are no female offspring, by that point, the jurisdiction had switched from FDA to EPA. And so the EPA assessed this particular application and carried out the full risk assessment for this mosquito. But in terms of the safety profile, what the FDA concluded was very similar to what the EPA has concluded for this particular project in Florida as well.
Great, thank you, Nathan. We've got 2 more questions. Oh, actually, we might have 3. But let's see. The first one, Kevin, we will send to you. Have any independent bodies, governments, universities, or others reviewed Oxitec's technologies, and are there any concerns on the human health front? Oh, Kevin.
Hopefully that's better. Thanks for the question. Yes, absolutely. All of our studies, when we go out in the field with these mosquitoes, are international, if you like. And as such, we only complete those in partnership with people who are stewarding those projects through, if you like. So there's not only collaborators on the project who have a vested interest in understanding whether this kind of technology works from an independent perspective, but there's also regulators who oversee each one of those studies as well. So there's universities such as the University of Sao Paulo in Brazil, which we've worked very closely with. There's institutes such as the Gorgas Institute for Human Health in Panama, a very prestigious institute who were independently reviewing the technology. We have people like the Gates Foundation who've looked at these technologies and decided that they would like to bring those along for things like prevention of malaria transmission. We have other academic bodies. Pasteur Institute has done reviews, and as I mentioned, all those regulators that Nathan was talking about earlier, either at state level or national level, or international level, when it comes to some of the large organizations, many of them have done independent reviews of our technology and our data post trials. I think it's been said already that we've released a lot of insects and over a long period of time now, and we've never had one human health or environmental health incident or adverse effect noted in all of that time. and lots of oversight and scrutiny all the way through. And as Nathan mentioned, each one of those studies has that post-release monitoring that carries on that monitoring right the way through till they disappear from the environment. Yes, lots of oversight, lots of independent validation or scrutiny, and not one adverse effect.
Thanks very much, Kevin. We're at time, but we're gonna take a few minutes to just do 2 more questions, and then we'll wrap up for the evening. And Kevin, the next question is for you also, and the question is: during use in other countries, have the Oxitec mosquitoes been found in areas a long way from the release? How far do they really travel in the wild asking for a friend?
Great, good question. So, every environment differs to some extent in its architecture and its landscape. And so these mosquitoes are pretty short flyers. They're focused around humans. That's their natural habitat. And typically they don't fly very far. So anywhere between just a few meters and round about 200 to 400 meters for rather extreme cases is the distance that they'll fly and disperse naturally by themselves. There's always the prospect with mosquitoes and other insect pests, pests generally, that they'll get transported inadvertently by humans, for example. And that's how mosquitoes often travel around the world and how Aedes aegypti is spread in many cases. But in terms of its natural dispersal, it's typically between up to about 200 meters, but you can get slightly longer distances up to about 400. Averaging typically at 50 to 100 meters.
And Kevin, for our US audience, if we were going to translate 200 meters to football fields, would that be 1 or 2 football fields?
Okay. 2 football fields, yeah.
Perfect, thank you. Okay, our last question, Nathan, is for you. And the question is: can you talk about the endorsement that the World Health Organization gave for Oxitec's first generation mosquito a few years ago? And can you talk about the recent publication by the WHO recommending that governments evaluate GMO mosquitoes?
Yeah, okay, thanks for the question. So in 2016, you may remember that was the year when the Zika crisis was really raging, particularly in Brazil, but also a lot of concern about the spread of Zika to other places. That's also when there were many cases of microcephaly in babies that were born to pregnant mothers that had had Zika infections. And so this was a major public health concern for the WHO. And at that point, they were looking for any tools that might be helpful in combating the mosquitoes that were spreading that particular disease. And so as part of their response to that, they endorsed Oxitec's mosquito, as well as a number of other new technologies for pilot projects to be able to control the Aedes aegypti mosquito, recognizing that a lot of the existing tools were starting to fail. Very recently, just a couple of weeks ago, the WHO has made a new position statement about the use of genetically modified mosquitoes to control vector-borne diseases. And they, again, make the statement in the context of the major impact of mosquitoes in spreading disease. And they are recognizing that new technologies, including GM mosquitoes, are a supplement or an alternative to existing interventions to help reduce or prevent disease transmission. So the WHO really thinks that any potentially beneficial new technology should be investigated to understand how useful it's going to be in the continued fight against these mosquitoes. And they're also urging that as part of the evaluations of these technologies, that the regulators are looking at the potential health benefits in consideration of disease control and not just looking at environmental risk. And that's an interesting shift in terms of how these sorts of technologies are assessed by the WHO as well. So this is very encouraging. It's very good news for Oxitec. It's also very good news for some of the other approaches to using genetically modified mosquitoes to control diseases in parts of Africa, for example. And so we're very encouraged by the endorsement of the WHO in this particular context as well. Yeah, I hope that answers that question.
Great. Thank you, Nathan. And this is the end of our webinar for this evening. And we appreciate everybody taking part. And we hope you'll join us on November 18th for the next one. It should be a really good tour through our labs that you'll get to take virtually from wherever you are. But hope to see you then. Thank you.